Sorry. Okay. So my topic today will be on transcript technology in gene amplification. I'll be talking to you about some technologies and platforms provided by Transgene Biotech. So Transgene Biotech is our main supplier who provide all our molecular biology reagents. Okay, so they cover from DNA or RNA extraction to qPCR to Western blotting to cell culture. So they do have a complete set of molecular biology reagents. So for your information, we are the sole agent for Transgene Biotech in Malaysia. So if you have any inquiries on Transgene Biotech, you can just contact us. And by the way, I'm Mr. Cheong. So I'll be conducting this session today. All right. So here is the two main technologies that I will talk about today. So I will start with Transo Plus. So this is a technology used in RNA extraction that will give you high yield and high purity. The second one is a transcript. So this is another technology for cDNA synthesis, where you can apply high temperature in your reverse transcription process. And for the last part, I'll talk about Transgene Biotech. So you will see how they are the one-stop solution from viral nucleic acid extraction up to detection level. So this part will focus more on COVID-19. Okay, so we start with this simple flowchart. So if you are dealing with DNA, you have to start with DNA extraction. Then you can straight away proceed to detection level. You can either do normal PCR, gel-based PCR, or qPCR. Okay, and after you do gel-based PCR, you can proceed to gene cloning. So our focus today is on our focus today is on this RNA part. So we need to do RNA extraction first. Then we have to convert them to cDNA. So this is the main difference between DNA and RNA. So after you convert to cDNA, we also call it as reverse transcription. Then we do mRNA level detection using either PCR or qPCR. Okay, so now we start with RNA extraction, which is this Transo Plus. So as you all know, guanidine method is a very popular method in RNA extraction. It has been used since many years back. Okay, so guanidine can help you to lyse the cells and help you to extract the RNA. So here are the list of the kits provided by Transgene Biotech using guanidine method. So this is why you see here you can see that guanidine method is so flexible and so robust that most of the kits here are using this method for their extraction. And we also have seed-type method, which is more on plant samples. So our focus here today is Transo Plus. Okay. So this is a simple evolution from Transo to Transo Up and then to Transo Plus. It's a basic upgrade. So Transo is actually similar to the Triso, where you always hear. Okay, so Triso is actually a reduced reagent for you to extract RNA from cells or tissues. Okay, so they combine this phenol and guanidine. All right, you just add these two in, and then after you do centrifuge and the lysis, so the RNA will remain at the aqueous phase here, and they will have 3 layers. Okay, so the pink layer is called the organic phase. So what you want is the what we call the transparent phase here, this aqueous phase. So all the RNA will be here, and this is the basic Triso. Okay, so Transgene have upgraded this Transo to Transo Up. So Transo Up is an improved version of Transo. All right, it has a more powerful lysis capability. So Transgene never stopped at Transo Up. They even further improved them to Transo Up Plus. Here, they combine the Transo Up with the spin column and washing buffers. So this is we call it a complete set, okay, of extraction using Transo Up and also purification using the spin column and washing buffers. So Transo Up Plus RNA includes two simple steps. The first step is liquid-liquid extraction. So we just lyse our sample using the Transo Up solution, and then we add chloroform. So after we centrifuge them, the RNA will be separated into this upper aqueous phase, as I mentioned earlier. And we transfer this aqueous phase into another new tube. And then we move on to the second step, which is the solid purification, where we involve the spin column. So here we just add ethanol to precipitate our RNA, and then we just wash them with these two buffers, namely the CB9 and the WB9. So in less than 1 hour, then we can get our RNA. Okay, so this is a result comparison. Here is sample T. Sample T is using Transo Up, while sample P is using Transo Up Plus. The Transo Up is without spin column, and Transo Up Plus is with the spin column. So as you can see, the yield of the of Transo Up is definitely higher because it doesn't go through the spin column. But from here, you can see that Transo Up Plus provide very good quality of RNA with higher purity. So this is what we actually want. So this is an example of the result that our customer provide to us. So actually our customer is working on RNA sequencing, okay, or transcriptome. So what he needs is a very good quality of RNA. So here you can see that the RNA integrity number that he got is up to 9.2. So with this quality of RNA, he can proceed to his RNA sequencing without any problem. So for those who are planning to do RNA sequencing, so this is a very good kit for you. Okay, so I'll summarize this Transo Up Plus RNA kit. So it can be used for various types of samples. For example, bacteria, plants, any cells or tissues. So basically, most of the samples you can use our kit to extract your RNA. This is due to the, it has a very powerful lysis. It will give you high yield and high purity. And the extraction can be completed within 1 hour. So it's quite fast. And then most importantly is this pink solution for easy visualization. So with this pink solution, with this organic phase here, you can easily extract this aqueous phase out without interfering with the organic phase, okay? All right. So after we do RNA extraction, we will proceed to cDNA synthesis, where we call reverse transcription. Okay. So Transgene has made some modification, okay, to their RT enzymes, okay. So they have 3 different series. The first series is EasyScript, Transcript, and Transcript 2. So the difference here is by different modification made. So the first modification made by Transgene to improve this enzyme is at the RNA's H domain. So with this modification, our enzyme will be free of RNA's H activity. So later I will talk more about RNA's H activity and what is their effect. So the second improvement is made at this DNA polymerase domain, at this part, the green part. This is to improve the amplification ability. So this modification is only applicable for transcript and transcript 2. As for the last modification here, it's only applicable for transcript 2. So this is to improve the thermostability of the enzyme. So I will also talk about this later on. So now we talk about RNase H minus. So RNase H is actually an activity. RNase H activity will actually degrade your RNA template. It will cut your template into fragments. So if you have your RNA fragments, you won't get your full-length cDNA, okay? So RNase H minus is an RNA-dependent DNA polymerase. So transgene has incorporated RNase H minus into the enzyme to prevent RNase H activity. So without RNase H activity, you will get full-length cDNA. So this is very important for you to get good results during qPCR. Okay, so let's think about this. If you have a RNA template with high GC, or we call a complex structure, okay, so how do we break open the chain or break open the complex structure? We need a very high temperature to release the structure, okay? So we might need to increase our temperature up to 50 degrees or 65 degrees. But if we increase the temperature to this high, to 50 or up to 65 degrees, our enzyme, our RT enzyme may be denatured, okay? So in this case, our enzyme need to have very good high thermostability. So this is the advantage of having high thermostability in your enzyme, in your reverse, in your RT enzyme. So this is a complex structure that I talked about earlier on. Sorry. So this red color loop, all right. So if let's say we proceed with 42 degrees Celsius, our primer will only amplify the green part, okay? And we will stop here, okay? So in this case, you will not get your full-length cDNA. You only get part of it. So in order to break open this structure, we need to increase our temperature up to 50 degrees Celsius, okay? So after we break open the complex structure, then only we can get our full-length cDNA. So in order for this reaction to proceed smoothly, our enzyme need to have very high thermostability. So transcript 2 is an example of one of the reagents that contain this high thermostability. So it can work between 42 degrees to up to 55 degrees Celsius. So when you face some problem with high GC or complex structure, as I mentioned earlier, then you have to increase the temperature up to 55 degrees to break open the structure. So from here, you can see that for transcript 2, it allows you to amplify your template up to 15 kV. So it's quite long, and then it has the highest sensitivity and fidelity, and it can work at very high temperature and also work on very complex structure, okay? Okay. So this is another upgraded version of transcript 2, which is transcript uni. So let's say if you have a template where even transcript 2 couldn't amplify, okay, it has a very, very complex structure that even at 55 degrees Celsius, the complex structure couldn't be released. So this is where transcript uni comes in. And transcript uni it allows your reaction to increase the temperature up to 65 degrees Celsius. Okay, so for transcript uni, it is only a 30 minutes reaction and only require 5 seconds of inactivation. So here they have also the RNase H activity free feature, and also you can amplify your template up to 20 kb. So from here you can see that there's a very special feature, which is this one-step gDNA removal. So what is gDNA removal? So gDNA is genomic DNA which we do not want when we are dealing with RNA. So we need gDNA removal to eliminate all the genomic DNA that we do not want. So usually what researchers do is that, or some other kits that you use, usually this genomic DNA removal is performed after extraction or during the cDNA synthesis. So it requires you to do 2 steps differently, okay, separately. So our special feature here is that we combine these 2 in 1 step, which means when you add your extracted RNA, okay, into the tube, into our reagent, you can it will remove the genomic DNA, okay, and at the same time, simultaneously, it will convert them to cDNA. This is because the gDNA removal will only target your double-stranded DNA. It will not interfere your single-stranded RNA. So it requires 15 to 30 minutes to have the cDNA templates done. So this is what we call do more with less. Okay, it is a 2-in-1 step. So still with cDNA synthesis, okay, we have another special feature by Transgene, where they add this anchored oligo DT primer. Okay, so traditionally for our oligo DT primer, okay, so we know that our template has a very long poly A tail. Okay, it could be up to a few hundreds. Few hundred base pair long, and then if let's say our oligo DT primer binds randomly at the A tail, okay, somewhere here, so this will reduce the efficiency. Okay, you will not get full-length cDNA. So in order to overcome this problem, Transgene has incorporated anchored oligo DT primer, where they have added AGC tail, okay, at the oligo DT primer. So they have, with this AGC tail, it will always bind to the 5' end, okay, of the mRNA. So if let's say your first base is U, A here will bind. If your first base is C, G will bind. So with this feature, it will increase the efficiency of your cDNA conversion, and at the same time, you will get more full-length cDNA products. Okay. So this is the result comparison using transcript tool, which is lane number 1. Okay, and lane 2 to 6. Okay, 2 to 6 is using transcript uni at different temperature. So here we can see that using transcript tool at 65 degrees Celsius, it will greatly reduce the background and also increase the sensitivity. All right. Okay. So in conclusion, with the presence of the RNase H minus, as I mentioned earlier, with the gDNA removal and also the what we call the anchored oligo DT primer, we will get a very good cDNA result. Okay. All right. So moving on to the last part. So Transgene Biotech is actually a company that provides a one-stop solution from this viral nucleic acid extraction up to detection level. So they have manual or automated extraction, okay, And also they also provide this multiplex probe real-time PCR or RT-PCR. So for the manual kit, they have this Easy Pure and also Magic Pure viral DNA RNA kit. So the difference here is that for Magic Pure, they are using the beads method. Okay, they are using the beads, while Easy Pure is using the spin column method as what you are used all the while. So Easy Pure viral DNA and RNA kit is actually provided to KKM. Okay, so we have been providing this kit to many hospitals in Malaysia for COVID-19 testing. So these two are the manual extraction kits, while Transgene has come up with automated nucleic acid extraction as well. So for TS32, this is an automated extractor where you can run 32 samples at one time, while for TS96 you can run 96 samples. For TS96 is even better than this one, because for TS32 you can only use Magic Pure viral DNA RNA kit, while for TS96 you can even use kits from other brands as well. Okay. So here you can see that for TS32, this is a qPCR amplification curve using this PRV. Okay, it's a pseudovirus DNA extracted from 32 repeat samples. So here you can see the consistency of the result. Well, for TS96, this is also another qPCR result using 96 repeated samples. Okay. Okay. So after you do the DNA RNA nucleic acid extraction viral, so you will do detection using real-time PCR. So here are the reagents that they provide. Okay, so they have the probe PCR supermix or even the one-step Q RT-PCR supermix. All right. So for the for these kits, okay, for the manual method or even the automated method, we do have a promotion now. So if any of you are working on COVID-19 testing or any viral samples, you can contact us, because for now we have a 25% off promotion. Okay. All right. So Transgene, as I mentioned earlier, Transgene provide a full range of product for molecular biology, from PCR to cloning, gene expression up and nucleic acid purification. So if any of you are interested to look more into this, you can go to our website at nanogenesolutions.com, and they will divert you to this transgenebiotech.com for more information. Okay. So here's another promotion provided by our company. So we have this research grant reward where you will be rewarded certain amount of grant if you make some minimum purchase from our company. So to know more about this, you can contact any of our personnel here. All right. All right, so here's, thank you for your kind attention. And before that, as I mentioned earlier, we do have a series of webinar coming on. So next week, we will have another seminar, which is on genomics and transcriptomics on next Monday. And the following week, we have another webinar coming on, which is on cloning. So here you will learn about our cloning platforms and the hands-on guide. So this is in collaboration with Monash University. We have two speakers or lecturers from Monash as well. So you can either scan here or you can register with the link that you received through email previously. you can just register anytime, and we will send you the link before the webinar starts. So, any question? I think I can unmute you all. Okay, any questions, you all can write it in the chat. Okay. Any questions? Okay. So, oh, and by the way, we are providing the e-certificate to all our participants. So please feel free to email to ngs@nanogene solutions.com. Okay, I hope you can see this email, ngs@nanogenesolutions.com. So please send your information, your full name, okay, to this email, and then we will send you an e-certificate soon. Yep. Any questions from anybody? Okay. Thank you very much. Okay. If no question, yeah, can you share your slide with us? Yes, of course. So if you need the slides as well, you can just write in an email to us, and we also will actually upload the videos of these webinars onto our website soon. Yep. Okay. So thank you very much, and I hope to see you again soon for especially for next week's seminar. Thank you. Hi. Did someone say hi? I'll be still here if you have any questions. Thank you. Oh, can you briefly explain about DNA extraction? What kind of sample are you dealing with? Oh, you mean the stages? You're dealing with plant samples. Do you mind if I share a protocol of our plant DNA extraction? That it will be clearer to you because we have the step-by-step guide. Please write into the to the email above. Thank you. Can your products be shipped to any country? Yeah, based on the inquiries, you please write into us, then we will make the arrangement. Oh, okay, no problem. Thank you. All right, so I think I'll end the session today if there's no other question. Thank you. Sorry, when's the next session? Next Monday. Please register yourself if you're interested in the next seminar, the next webinar, sorry. Did you receive, how do I register? Did you receive the email? Okay, you can register here. Okay, here. You can scan the QR code. Okay. All right. So here's the link. Okay. Thank you.